Hello and welcome to Virtual Regeneron ISEF. I am Anna Runs and I manage our Science News in High Schools program for the Society for Science in the Public. This program is one of the Society's outreach and equity programs and brings our award-winning science news journalism and related educator resources to almost 5,000 schools this year. In 2015, the Society founded our equity and outreach program, ultimately aimed at expanding access to authentic STEM experiences for underserved students. To accomplish this goal, we support adult educators and mentors with grants, conferences, resources, and a community while running our research competitions like ISEF in parallel. As you're all aware, conducting research this summer might look very different for many students. So in this session, titled Research at Home Life Sciences Edition, two expert high school research teachers from both our outreach and competition communities will convince you that authentic research can be done anywhere and even from home. Together, they will share examples of model organisms their students have successfully worked with and explore new possibilities for mentoring and supporting at-home student research. As a former high school science teacher, I am thrilled to introduce our teacher speakers today. They have both presented and received rave reviews at our Research Teachers Conference and have even judged in our competition. Once introduced, the speaker will tell you a little bit more about his or her background and experience teaching students research. So we'll start off with Dr. Tina Gibson, who teaches at the Mississippi School of Math and Science in Columbus, Mississippi.
So good afternoon, everyone. As Anna said, I am a biology teacher in Mississippi, but I've also taught in Kentucky and in Florida. I have also been teaching a research course for over 20 years. I was a fair director in Kentucky for several years, and I've also served as a judge in the environmental sciences and plant sciences at ISEF. And this will be my 18th ISEF event this year.
Thank you, Tina. And I'm also very excited to introduce my former colleague, Dr. Bill Wallace, who teaches at Georgetown Day School in Washington, D.C.
Thanks, Anna. Previously to teaching, I was a research neuroscientist conducting research and directing labs at the Mount Sinai School of Medicine and at the NIH. I was interested in looking at the molecular biology of the brain in response to injury. For the past 22 years, however, I've been teaching at Georgetown Day School in Washington, D.C., including a neuroscience course, physiology, and most relevant right now, a biological research course in addition. I have been associated with the Society for Science in the Public for many, many years, including being the inaugural head judge for the Broadcom competition.
Great. Thank you both again for agreeing to be here and share your expertise and also how you're pivoting to support student research this summer. We really appreciate it. So now I'm going to hand it over to the presenters for about 25 minutes. And then after that, we will have a 15-minute question and answer session. So audience, please submit your questions via the chat throughout the presentation, and we will be collecting and queuing up as many of those questions as possible. So Tina and Bill, the presentation is yours.
Okay, thank you. I'll go ahead and start off. During this recent COVID-19 pandemic, scientists, professionals had to adapt to the worldwide quarantine. They have not been able to go to their laboratories as usual, but what they have done instead is brought the laboratory home. And what we'd like to convince you today in this talk is that students, high school students, can do the very same thing. So Tina and I are going to give you some examples of what research has been done in our classes, and hopefully we can convince you that sophisticated, authentic research can be done at home if you follow a few simple rules. Next slide, please. Slide. An example of that can be shown here, where we've used pill bugs in our research class. Pill bugs are creatures that you can find in your backyard or your front yard, not difficult to attain. They can be bought also, but they become a wonderful model for learning and memory in our class. They have a very simple nervous system, but they can be taught by classical association to change their behavior. This opens up wonderful possibilities of using a very simple organism to study what goes on within the brain of a pill bug while it's learning a new behavior. Slide. But what does it mean to actually do authentic science research at the high school level? Well, it's not much different than the sort of research that I did in my laboratory at Mount Sinai or at the NIH. It's hypothesis driven. That is, that the student is not looking for repeating results that other people have done, but are rather conducting open-ended investigations where there's not a correct answer that's known ahead of time, but rather that answer depends upon the hypothesis that was developed. This is exactly what scientists do. In addition, the investigation is actually a series of experiments where the results of one experiment will suggest the next, as opposed to the more common in high schools where you do one experiment and then you're done. So even though you're doing sophisticated, authentic science research, what we really want to let you know is it does not require high technology. Instead, what is important is that you are using an appropriate model organism. This is the biggest difference between the possibilities you can do working in a laboratory, let's say at a university or research center, and what you can do at home. Now, that doesn't, although you may not be able to use the most sophisticated animals or plants, the organism that you use must match your scientific question. So for example, if you're interested in understanding the genetics of behavior, fruit flies would probably be a better model organism. On the other hand, if you're interested in studying cancer and cell proliferation, planaria might meet the needs far better. So a lot of thought should be going into the animal model system or the plant model system that you use for the questions that you're asking. Obviously, the model's got to be practical for experiments at home so that you can culture those animals, you can maintain those animals or plants, and you can perform the experiments on them. The experiment must yield measurable results. That is, just like in all science, you need to be able to quantitate your answers. So for example, if you're looking at behavior, it's important to put that behavior within some kind of a number. And in addition, we want to emphasize that all of these simple model systems we're talking about have an extensive scientific literature. So not only were they simple, makes it convenient for work at home, but they're simple and scientists have taken advantage of that to be able to ask difficult questions when they have a simple model system. Next slide, please. So Tina and I are now going to give you a few examples of such model organisms. I'll talk about animals that we have used in our research class at Georgetown Day School, and Tina's going to talk about some examples of plants. So some of the animals that we have used with good success are these pill bugs. They're also known as roly-polies, perhaps, planarian, fruit flies, bean beetles, and brine shrimp. Now, I don't want you to limit your research to just those organisms. There's lots of other possibilities, but it gives you the idea of what the capabilities of these animals are and the sorts of experiments that you can do. So Tina?
Okay, so for plants, students have used in the past, they've used scallion onions, we've used red peppers, some sort of citrus plant, a legume, Wisconsin fast plants, and herbs. And just as Bill said, this is not an exhausted list. There are many other plant samples that you could use. Next slide, please.
So let me give you some examples of how we've used these animal models in our classroom. For example, as I had mentioned in a previous slide, pill bugs are fascinating to study for learning and memory. And our students made this really interesting discovery that we can teach them a new behavior using classical association, the type of experiment that you might be familiar with using Pavlov's dogs. The fact that they can do that opens up possibilities now that you can study subtleties about the learning and memory that these pill bugs exhibit. Another example are bean beetles, and I'll explain later how bean beetles can investigate resource partitioning or ecological resources. bean beetles are very useful because each female is attached to one specific bean. By changing the ratio of the female to the beans, you can ask questions as to what happens when the resources are limited. Next slide, please. So two other examples are going to be the planaria, and you may be very familiar with the incredible regenerative properties that planaria have. So they are a great study for cell proliferation and differentiation, and apply that, for example, to cancer or to wound repair. And of course, the classic fruit flies that can be used both in terms of developmental biology. In the larva form, you can change their environment, or you can study their behavior from a genetic point of view because they have such a short generation time and there's so much genetics that's known behind it. Tina?
Okay. All right, so on the plant side, for onion cell, excuse me, regeneration, you could use scallion onions, and that can be conducted by using various household products. You can also use plant hormones that can be purchased very easily, and measure root length. If the student were to choose to go to the next level, they could actually look at that, the mitotic division, and comparing that statistically. Next slide, please. Using plants for regeneration as far as tropism or gravitropism, that would be really good for places where planting might be quite difficult. Understanding the effects of that could actually make or break a crop. Next slide, please. Plant physiology. This is one area that my students find very interesting. And so what we do is we take a slide, we take type of monocot or dicot leaf, paint it with clear fingernail polish, and use tape and pull the stomates directly off of the backside of the leaf. And we can look at that. And this would be very good because then they would be able to look at stomate function in various climates. And that would be very applicable to what we're going to talk about in just a little bit with the Sustainable Development Goals. Next slide, please. So seed development. As I mentioned before, the Sustainable Development developed by the United Nations is something that we're really looking into as far as drought tolerance. And so you can use turf grasses, you can use yard grasses or crops, because even if the seed is healthy, if the seed cannot survive the drought situation, then that's going to cause a challenge for the future generations to be able to grow these crops. Okay, next slide, please.
Okay, so even though these are pretty sophisticated experiments that we're going to be talking about, and you can get some pretty sophisticated types of results from it, I would really like to emphasize that you really don't need to have very complicated apparatus. And in fact, for many of the experiments that we're talking about, our students fabricated or adapted very common materials in the lab to be able to perform these experiments. A couple of examples I'd like to present to you now. For example, with the pill bugs, we wanted to have a choice chamber where the pill bug could choose to be in one environment or the other environment, and then to change its choice based upon learning and memory using classical association. Well, fancy stuff, but basically all it is two Petri dishes that are connected together so the pill bug can choose to be in one or the other. And your counting is, well, your eyes. So you simply count the number of pill bugs in each chamber. So that's very straightforward. Similarly, if you want to do something like this with fruit flies, one of my students put together an apparatus, which is simply two 2-liter bottles of Coca-Cola that have been cut in half and connected with PVC piping, and then the flies are allowed to go up through the PVC piping and then make a choice into one chamber or the other, depending upon what you put in either. The beauty of that is you can then capture the flies in one chamber or the other chamber, breed them separately from each other, and then ask if their offspring show the same behavior. In other words, you're asking the question, was that choice a genetic choice? And I think that was some fascinating experiments that have been done by some of our students. Tina?
Okay. So for plants, you could actually boil down these plants to extract the pigments and use them in chromatography studies. I've had students that have actually taken indigenous plants and looked at them to make types of gels, maybe something to treat burns or something in the future. And another very classic study would be using the Wisconsin fast plants, very simply placed on a paper towel, put in a petri dish, and you can look at various genetic studies using these plants as well. Okay, next please.
Another thing that may be overlooked is the fact that your cell phone can actually be a laboratory apparatus. That is that you can get wonderful pictures using the phone, the camera on your cell phone for the photomicroscopy. So here's a couple of examples of a regenerating planaria, the head portion or the posterior portion of that planaria, and the images are perfectly fine for the research that you might want to do. We have had a couple students in the lab recently make a fascinating discovery that if you stop cell proliferation, what happens to the planaria is they actually undergo apoptosis. And we're exploring that right now. Next slide.
So for this slide, this talks about, and as well, and then I'll reiterate what Bill just said with using your cell phone. These were both taken by a cell phone using on the microscope. And so students looking at a basic experiments of red onion cells with insult solutions and looking at plant stability and how much tolerance they can take in as far as salinity is concerned. And this was taken before and after plasmolysis. And this can be very beneficial, especially for us that may be close to the coast and the salinity that may be happening with those coastal line plants. Next slide, please.
So Tina and I now want to just give you some interesting experimental results that we've gotten using these very simple model systems, using sophisticated thinking, but simple apparatus. For example, and I returned for the last time to our pill bugs. So I've talked about the idea that they have made choices that are flexible depending upon how they're taught. The results would look something like this graph that you've got here now, which is a pretty sophisticated graph. Behavior generally does require lots of numbers of pill bugs to take a look at. And in this case, the bottom line represents the behavior or the choice of the pill bug before it was trained, and the top line represents the pill bug after it was trained. And you can see that the lines diverge from each other. That is, that after classical association, the pill bug literally changed its behavior, and which is a definition of what memory is. However, I want to emphasize that this result only becomes clear because of sophisticated statistical analysis. So the same sort of statistical analysis that you would use when you're in a laboratory, you would use at home. In this case, it was standard deviation, error bars, and student t-tests for looking for group differences between two groups of numbers. So this is a sophisticated result. Similarly, on the next slide is shown another result that was a very interesting discovery done using very simple apparatus too. In this case, fruit flies were raised in various amounts of ethanol or without. The larvae were then allowed to develop into the adult form, and the student took a look at the time it took for those wings to be developed once it became an adult and it could spread it. And they found a very interesting discovery, which was that in the low levels of ethanol, the flies took significantly longer, as shown in the dark bars on this slide, to spread their wings compared to the control, which are the light bars. This is a great example of how you can look at developmental biology using simple manipulations of the environment, for example, whether there's ethanol present there or not. And on my last slide, we'll go back to the bean beetle and an interesting discovery made by a group of students at doing research in my class, which was that they asked the question, if we limited the resources to the female bean beetle, how do they adapt to the limited resources? In this case, it was the amount of beans that were available that the female uses to lay her eggs. And as the amount of beans became less and less, fascinating, the female actually gained greater mass as the amount of beans that became available were less and less. To my mind, and to those of the students, this is an original discovery, as were the other three that we just talked about. And given time, that would be a fascinating experiment to follow up on to build it into a full-fledged investigation. So those are three examples from the research that we did using animals. Tina's now going to talk about how you can use plants.
Okay, so as I stated earlier about the Sustainable Development Goals, that's a really great place to start investigating topics. And because plants are so vital to the world food supply and such, students can get on there and research, and they can maybe make an impact on a global perspective that may be outside their town, their state, or their country. Next slide, please. So the next examples that I'm going to show are ways that my students look at helping with when doing studies with plants. And so math modeling is a very hot topic at our school right now, machine learning, and being able to use resources that we have locally in Mississippi. And so we're going to look at a pollution analysis and that effect it might have on agriculture. We're going to look at a tilling project with agriculture and also a lichen study. So this first project, this student has taken open source data, and this open source data was retrieved every 6 hours over a time span between May 15th and May 2015 and April 2019. And so taking that data every 6 hours and giving fresh data, they were able to extrapolate that information to look at pollutant clusters that might occur by the year 2024. And this can be a great benefit for being able to look at statistics of where our pollution may be heading. Next slide, please. So this slide is looking at agricultural tilling practices. And so this student was able to use machine learning. Again, all these numbers were taken from open source databases. And so they're going to map this agriculture tilling practices with airborne sensing imagery. And once they do the sensing imagery, they're able to use computational cost to look at improving tillage efficiency, which is of utmost importance in agricultural states. Next slide, please. So this last study is looking at lichen. And so the student actually retrieved a couple samples from Alabama and several samples from Mississippi. And so what she did is she went through and retrieved these pieces from across the state, took them and used different types of solvents that you could get at home, such as isopropyl alcohol, acetone, and even water, and bought a pH buffer. And so a really good first level project would be to be able to extract these pigments and see and look at their results from that. She was also able to apply them to the Sustainable Development Goals, and for those that are in developing countries that may not have very sophisticated resources. And a good second phase of this project would be to take this project, take it to the next level, look at its antimicrobial, which is what she was able to do. Next slide, please. Okay, so we've covered several topics now that what students could do. And so what I would like to talk about for just a few minutes is how you get those studies, how you get those ideas. And so here in Mississippi, we have lots of local farms. We have 20 million acres of forest. So our paper mill is of utmost importance in recycling. And so using those resources to find that topic can be very important. Also, while teaching in Florida, I was exposed to an app called WeatherStem. And WeatherStem has these apparatus planted across the United States, and you can go in and look at soil moisture and water temperature, which it actually could affect agricultural processes as well. And the last part, looking at water analysis, collecting those samples, looking at the streams, looking at algal blooms, the plant ecology, all these things students just need to get out and look to investigate that particular area, whether or not erosion is happening, is it the salinity, Is it the invertebrate biodiversity that's being disrupted? And so very many ecology projects that are dealing with plants and such could be developed. Another really good place, which is not on this slide, is also looking at science news. My students spend a few weeks going through science news in the classroom, getting ideas and being able to springboard off of those ideas. Next slide, please. Okay, so now we're looking at how do you establish this partnership? So through universities, being able to look at a lending program, which Phil is going to address, those supplies, being able to go out and talk to your local farmer and being able to interview. And as I discussed on the next slide, we want to look at, thank you, the next slide, we want to do like a form letter. So what would happen, so if I develop, have a form letter, I send these out with my students, perhaps they can get these slides and they can decrease the cost. Society for Science research grants also are a great place to start. And again, going back and looking at these big corporations, your local businesses, and your big places like Nissan that we have here in Mississippi, all these places will help in developing these partnerships to lend for mentors or supplies, and just getting topics nailed down. Next slide, please.
So another way that schools can help in terms of supporting kids doing their research at home is an idea of using lending kits. That is, normally the student or the student would be working in a laboratory. You take for granted that you would have all the things that you will eventually need to be able to do the experiment. And then rather than force you to make your own test tubes or make your own microscope, what we'd like to see and what we're establishing at Georgetown Day School is a lending kit where we talk with the students ahead of time as far as what their research project is going to entail. And then what we're going to do is we're going to put together those supplies and those equipment that that student needs in order to run it at home. So for example, if a student is using planaria, we would make a dissecting microscope available for that student. But you can't overlook just the common sorts of things that every student needs: a pipette, pipette tips, salt solutions, test tube racks, filter paper, all those sorts of things. So it's sort of like a care package, but it's a customized care package that each student would require to do their work at home. So hopefully schools that you go to and students can make such a partnership where they can get these sorts of supplies that will facilitate the ability of them to be able to conduct these sorts of experiments. So you don't have to build every piece of equipment. Could I go to the next slide? Okay, so what hopefully Tina and I have been able to do is to convince you that you can indeed conduct authentic biological research at home. Even though you have the limitation of being at home and not going to a laboratory, you can basically mimic what professional scientists are doing when they're taking things to home when they can't work in their lab. And it follows the same sorts of procedures you would do if you were in a lab. So you want to ask good questions and you want to follow your interest, your passion, whether it's genetics, whether it's cancer research, or whatever, you can continue to study that. You want to make sure that you choose an appropriate model system. So you want to think about the question you ask and then ask which model system would fit it. Fruit flies, onion plants, or whatever. You don't need fancy equipment. So you can use simple and available technology, either building on your own. Online, you can probably find examples of people who have made simple equipment, or what you can borrow from your school. Importantly, even though the work is done at home, it's still, you want to obtain quantitative data and use the same sort of sophisticated statistical analysis that you would use if you obtained that data in the lab itself. And hopefully that you can do that with the advice from a mentor or mentors, which you can do online. Most importantly of all, you're going to use scientific thinking. And in fact, the challenge of doing research at home really puts the onus on you to be able to think in scientific terms. And I think that that will be very impressive for the judges, that you are able to be resourceful enough to do your research at home. So I hope that these slides helped. I know that it's not complete, and we would love for you to have more feedback. So if you have any questions, please feel free to either email me on wwallace@gds.org or Tina T. Gibson at the msms.org and ask us if you have any questions or for protocols and such. And in addition, on the SSP website, we've included some additional resources we didn't have time to go into that you might want to avail yourself for. So thank you very much for listening to it. We hope that this was helpful to you, and we very, very much would love to help you guys do the same sorts of research that we've had the good fortune our students to do.
Great. Thank you so much, Bill and Tina. You guys gave some incredible examples for at-home research ideas today. So I'd now like to switch over to our question and answer period. We have some questions coming in from the audience right now. And audience, if you, again, have additional questions pop up, please put them in the chat box. The first question you all spoke a little bit about already, but maybe we could do a little more brainstorming on, is it possible to obtain some biological lab equipment cheaply? And the second part of that is, could we build some equipment? So why don't we go ahead and start, Tina, with you, and then we'll move on to Bill.
Okay, so I'm sorry, I'm getting back, I'm getting an echo. So yes, I do think as far as building the equipment, I think the biggest thing that you would have to look at is making sure that you're meeting the requirements. And so before any of your investigations start, I would definitely reach out and try to find someone that can look at what your apparatus is. I know my students in the past have actually emailed copies and pictures to various professors and just said, hey, does this look good? You know, is this something that looks feasible for me to test this particular topic? And they've been really receptive in saying yes or no, you need to make this, change this around a bit. And then again, I think as far as it's looking as far as the lab equipment cheaply, our school is looking into the lending process as well. We are asking for students to come to the school to pick that up and lend to them for a couple weeks. That way we can loan them the equipment and they don't have to worry about purchasing it. We just need to have that returned. What about you, Bill?
Yeah, a couple of things I'd add to what Tina says, which makes a lot of sense. You would be scared, surprised what sort of scientific equipment you can get on Amazon. So that's one source of glassware and things like that. Now, hopefully you don't need to do that, or the school can support you and do that. We don't want you to go broke doing your investigation. So that's one thing, is that there are resources available online. Second thing I'd like to emphasize, and this is based upon experience of my kids, is be patient. I've had kids build their own apparatus as part of their project. And what they do is they go through the engineering process. They try to build something that they think is going to work. They try it out. It doesn't work. You reiterate. You try to build again until you get what you want. So, for example, I showed you that picture of the two bottles connected together. The student that did that didn't immediately come up with that. It took him days to be able to figure that out. And make that part of the experiment, because by building that apparatus, you're really thinking about, well, what is it that I want to test? How is it that I'm going to test it? What's my dependent variable? How am I going to get numbers? So even something as simple as building these apparatus, you can go through the scientific process. And maybe what that means is it may take you a week or so to get the apparatus the way that you want it, but that's time well spent. At least that's been my experience with it. And as Tina mentioned, please, we've always found that scientists are great mentors, scientists, engineers, people that work in businesses. And you got to realize that these scientists aren't having students in their lab this summer because the labs have been shut down for the same quarantine. So they're probably going to be even more available than they were before. So I would say don't be shy. And if you find somebody that's working with fruit flies, sleep activity of fruit flies, there are a number of experts in that field. Feel free to ask them, what do you think? What needs to be in this apparatus? And my experience has been, and my students' experiences have been, that these scientists have been very, very collegial with our kids. And it's a great experiment because it mimics exactly what professional scientists do.
And to kind of ride on that with Bill, we've gone to our local university and they've actually given the kids certain types of plants to work with. And using that letter, that form letter that the teacher can send out just very generic has proved to be very productive. Even going to the local grocery market and saying, hey, I need 10 radishes, I'm going to do an experiment, or I need X amount of different types of flowers, have been very receptive in donating those to the kids. So that could be another way of looking and getting those supply pieces.
That's great. Thank you both. So our second question from the audience is, and we'll talk a little bit about this actually in a session later on today, but where can we find open source databases? So Bill, why don't we go ahead and start with you?
Well, I would suggest Tina, who's had more experience with that. All I'll say, just real quick, is that there are lots and lots of websites for specific animal models. So there is a fly page, there's a planaria page that gives lots of information. There's also genetic stocks for various organisms that you can have access to. And if anybody has a specific question about animal model, please send me an email and I'd be glad to send you links to those sorts of sites. But I think Tina's probably had more experience and so can probably answer this question better.
Okay, so basically what my students did was literally call the university and say, hey, I want to do an agricultural project and I need open source data. Now, some professors were really good about saying, hey, you can just use the data that I've collected for X amount of years, or they've guided them towards that database. And I would like to reiterate what Bill said. Email me and I will give you the links of what my students have, because there's the Human Microbiome Project, which is an awesome site, and it's open source. And these students will use, there's a couple links at the end of this PowerPoint that actually have things with, I think it's called PhyCharm, which is for the extreme machine learning for kids to do programming. And so if you'll send me an email, I'll be more than happy to find as many links as I can from my students, what sources they use. But I would say a large portion of them talk to the local university, and then they guided them to say, hey, this is an open source. USDA, we have a very large department here, and our extension campus with Mississippi State, and they were able to help us out with finding that data that's already been retrieved. So send me an email, and I'll be more than happy to try to send out as many links as I can.
Sounds great. Thank you. And we will, again, have another research at home session called Big Data and Engineering this afternoon. So definitely tune in to that to grab a bunch of open source data websites as well. So next question from the audience here, hopefully we can do a little brainstorming again, is how can we mimic lab conditions, for example, temperature, constant temperature conditions at home?
I smile at this one, and I got a real quick story. When I was a postdoc, after you get your PhD, I postdoc in a very, very famous lab at Yale University, Nobel Prize winning lab, and we were just starting to clone. And you have to have set conditions, 37 degrees, to grow the bacteria, but the lab was a neuroscience lab. It didn't have anything. What we did is we literally bought a hairdryer, turned it on the highest we could, and found what position to put it in within a cupboard to make sure that cupboard was at 37 degrees. Now, later on, we got more sophisticated, but the point is you can adapt. And so what you want to do is you want to make sure you've got a thermometer. And you can use that thermometer and then look for places in your house or outside that have constant temperature. Behind a refrigerator. I know your parents are probably going to kill me for saying things like that. But feel free to adapt that you don't need to have a constant water bath in order to do this. You could literally take a tub, put ice in it, and the ice will keep it at a certain temperature for a set amount of time. But I think, again, if you have a thermometer, you have an incubator. You just need to find where in the house you can do that.
Yeah. And I would have to back that with Bill because I've used bathroom cabinets, kitchen cabinets, coolers, lots of styrofoam, like things that you get shipped. Styrofoam containers are fabulous. You can put a hole in the top. I use that for my fruit flies. I made my own little habitat for my fruit flies by putting the thermometer in the top and such, and then types of buckets if you want to insulate them on the outside. I've actually had my husband make something with PVC pipe and build little walls, so to speak, like curtains around it. But honestly, a cabinet works just as well. It just has to acclimate. And once it becomes that temperature that you want it at, a lot of things can happen at that point. So.
Yeah, to emphasize, you may not get it at exactly 37 degrees, but you'll know what the temperature is, and that's what matters.
Exactly.
That's great. Thank you all. I think we have probably time for a couple more questions. The next one is, are there opportunities for team research or internships now during lockdown, like online opportunities? And obviously, I don't know what experience you all have right now with searching and finding those opportunities, but do you have any in mind that you know of for maybe your students are working through?
Are you talking about as far as a team, for them to be able to work collaboratively together, or at I guess I'm not.
Yeah, I think potentially those are two separate questions, or we can answer them separately. Are there kind of collaborative research ideas that you know of that are online, and then maybe separately any other research ideas that you know of?
Well, yes, I do know of some research opportunities that are online. Unfortunately, many of them cost, but I have been posting them for my students for the last month or so. But again, send me an email and I'll give you all the links that I have for students to be able to do that. Almost everything this summer will be online experience, a virtual lab. There are many summer camps, even my own at our school for our 7th and 8th and 9th and 10th graders, we have made into distance learning where we're making a camp and then making all the lesson plans, and we're sending that out for students to be able to look at. So I would say that, yes, there are some opportunities out there. I'm going to be limited in what I have, but I'm more than happy to share it.
Yeah, and then I have two thoughts. Like probably a lot of you that are listening into this, my students were looking for internships too, and there's just none to be found. This quarantine has affected everybody. So they are pivoting to being able to do this research at home. The second thing about collaborations is most scientists collaborate remotely. That is, for example, when I ran a lab, I would collaborate with people that were halfway around the world. That collaboration is where you have a person that has an expertise in one way of looking at a problem, and you may have a different one. So you could have a math modeler collaborating with a biologist, for example. And so I think those collaborations can still survive this virtual world that we're living in right now, and it still mirrors what professionals do.
Exactly.
All right. Well, we're actually kind of rounding out our time here together. So I'd first like to thank the audience for joining us today and asking some really thoughtful questions to our presenters. And then Tina and Bill, on behalf of the Society and also the Regeneron ISEF community, we thank you for sharing your time, your expertise, and for supporting so many students, especially as they navigate these new and very different circumstances in doing their research. And then one more quick note, if you are a high school science educator in the audience, and if you're interested in learning more or signing up for the Science News in High Schools program or the Research Teachers Conference Lottery, please head to the Society website, or you can even go to the About the Society room on the virtual ISEF platform. Those opportunities are available right now, so please go for it and sign up. Thank you all so much for tuning in. We appreciate it.
Thank you. Thank you.